So welcome you back, my dear students. So in the last video lecture tutorial, we are talking about DNA and its mutation and its identification and the types of mutation based on the different sources and characters and many more things. So in continuation with the previous video, we are going to finish by today hopefully. Uh, the types of basically the source and the types of mutation based on the different conditions. So here based on the character you can see there are some mutation like based on the morphological character. Morphologically you can see alters morphological characters also known as visible mutation. So when the morphological changes is visible, that means you can see or you can easily identify in naked eye, so that can be termed as visible mutation. So see there are some examples: dwarf peas, short legged sheeps, curly wings in Drosophila. So these are the morphological changes and morphological mutation that you can see. Next is the biochemical. So biochemical mutation is generally cannot actually identify easily by visual observation or by naked eye because it alters the production of biochemicals. So in a particular plants or animals, what are the biochemicals like some biomolecules or any um, secondary metabolites, primary metabolites, their production will hamper basically. Okay, so that can be measurable once you extract them from that particular animal or plants or micro. Next is the based on visibility. Visibility means the mutation you can understand by observing or seeing or by visibility. This is also further categorized into mutations and micro mutations. So what does it mean by macro mutations? Macro mutations with large number of changes in the phenotype. So when in a particular population, large number of changes, suppose suddenly due to suppose a cold stress or heat shock, suppose a large population of a particular forest or a animal population suddenly changes their sleeping behavior, say. Okay, so this is one kind of morphological mutation that you can see. So it can be easily detected because it's a large population. Easily it can be detected, found in qualitative characters also. Found in qualitative characters. It's not the quantitative characters. Qualitative characters means their behavioral nature, their if it is in plants, their production, or if it is an animal, so the cow, the production of milk, that will be hampered. So found in qualitative characters. Okay. So here, in case of micronutrients, you can see mutation with invisible phenotypic changes. So mutation means you can see in the naked eye, but micro mutation means you cannot see, you cannot observe the mutations. Okay, so this is the invisible phenotype changes and not easily detected. Special techniques requires. So there are some special technique to detect the mutations. Maybe the mm, molecular and biochemical and some biophysiochemical techniques are there to identify the mutations and observe in quantitative characters. See the difference. So it is very important slide for you. So write the differences between micro and macro mutations based on the visibility. So these are the 3 points you can easily write and you can explain. Observe in quantitative characters because this is a very small number. That's why it just comes under the micronutrients. quantitative characters, and here it is the qualitative characters. Okay, so just go through it repeatedly. I hope you can understand. If you have any queries, you can ask. Next issue is the based on the survival ability. The mutation is actually categorized into lethal mutation. Lethal mutation kill all the individual lethal gene is there. Sublethal mutation, okay. So it ja, just a half harmful. Lethal is the full if it is considered the full 100% mutation. So sub means the little bit less. So kill most of the individual. Here is the all individual and most of the individual because less harmful. Subvital mutation kills some of the individual, so it's little less than the sublethal mutation. Okay, vital mutation is do not affect the survival. Okay, so because this is important, vital mutation do not affect the survival. Super vital mutation increase the survival. This is the opposite thing. Is the important side of mutation. See here, only the lethal mutation they generally kill the individual, but vital mutation or super vital mutation they actually increase or help to the survival. Okay, so this is the important thing. So based on the cytology means karyotype. Karyotype cytology, these are nothing but the study about the chromosome. So in case of human, twin equals to 46, where 44 chromosomes are autosome and 2 X and Y are the sex chromosomes. So based on the cytology, means based on the chromosomal aberrations or mutations, there are some mutation like the chromosomal mutation changes in either chromosomal number or structure. So during breeding, maybe some chromosome deleted and structural abnormalities may be found. So, particularly for that mutation, chromosomal mutation occur. Gene mutation, you know already that is a alteration in base sequences of one certain gene. So this is also due to the DNA replication error or due to some carcinogenic agent or UV rays or some chemical mutagens. There are a lot of chemical mutagens. Here it is also important cytoplasmic mutation. Cytoplasmic mutation means changes in chloroplast DNA, cpDNA, and mitochondrial DNA. So once the chromosomal mutation, it is nothing but the chromosome number in the nucleus. And here cytoplasmic mutation means mutation in the chloroplast DNA because it is in the cytoplasm, and mitochondrial DNA because all the cytoplasm contains chloroplast and mitochondrial DNA. Fine. So, based on the effect on the functions, so there are a lot of functions of the mutation. So, based on their functions, they have been categorized into several things like loss of function mutation. See, here, loss of function mutation. So, that means if any particular tissue or any sample, any organism is having the mutation, that means they will reduce the function. It is very understood. It's not so complicated. Gain of function mutation gain means getting the function of mutation. That means change in function. Okay, so function will be changed, and loss of function mutation means the reduced function. This is a very simple thing. Fine. So here I have already discussed in the class. You can see some mutants. The product of mutation is called the mutants. The morphological mutants detected by outward appearance of an individual, that is shape, size, and color. Lethal mutants cause mortal or lethal effect or killing effect rather on the organism. Conditional lethal mutants that expresses characteristics of the wild type when grown under certain conditions. Okay, this is a particular for the wild type individual. Biochemical mutants for bacteria, biochemical mutants need to grow in media supplemented with specific nutrients such mutants are called autotrophs. Sorry, auxotrophs. Okay, so auxotrophs are nothing but the mutant, biochemical mutant that is used to detect the mutation in bacteria. So this is the important thing you need to remember. Okay, so auxotrophs are the mutant, chemical mutant that can be useful to detect the mutation in bacteria. Okay, fine. So here are some mutagens also, like physical mutagens is already discussed. X-ray, gamma rays, alpha particle, beta particle, fast and thermal neutrons is also important. UV rays, chemical alkylating agent based analog, uh, acridine dyes, other mutation, nitrous oxide, hydroxyflavine. So these are the new things. So you can already remember what are the things I have written in the board. You have already noted down. So these things also you can include here. So this is the overall thing. So this is a little bit thing I can complete it here. So material used for treating with mutagen. Mutagen, if you want to check the mutagen, if it is in plant system, so you need the things like the seeds, pollen, vegetative buds, whole plants, bulbils, tubers, and suckers. And significance of mutations in plant breeding. See here, this is also a very important thing in case of plant breeding. When a desirable character is linked with an undesirable character, so you need mutation. So desirable character linked with the undesirable character. So undesirable character, this portion you need to stop. So you need to do the mutation so that that undesirable character stop doing function. Okay. To know source of resistance genes in the available germplasm. So because this is the germplasm is the gene bank. It's like germplasm means all the phenotypes of varieties where are cultivating on a regular monitoring. So there also, if you need a gene bank or resistance gene, you need mutation to create variability. So if you want to have some changes, differences, genetic changes, you need to create mutation. Otherwise, you cannot change the variability to develop male sterile line. So this is also so when this is your target thing. So male sterile line means you have to work with the sex chromosomes. Either you have to stop their function of the sex chromosomes. So that is nothing but the chromosomal mutation to create variation in vegetatively propagated plants. This is the same thing. Okay. So this is the importance of mutations in plant breeding. So in the next video tutorial, we'll start how mutation can be detected. Okay, mutation can be detected and what are the techniques. Today it's enough. So thank you for watching.